All right, we're going to take a look at the modification of mRNA. So this is another way that we can modify gene expression. So we've already talked about in DNA, so I'm going to go back a few slides here. In DNA, we have regions that are called exons and introns, and that's within the transcriptional unit, within the gene itself, right? So this whole thing is going to be transcribed into mRNA. But some of the areas are for coding the gene, the exons. Those are ones that are going to be translated into amino acids. And the introns actually will not be translated into an amino acid. They're going to be cut out and the exons will be spliced together. An intron, by the way, is another example of DNA that has a function that isn't, essentially going to be a gene coding for the amino acid sequence. So, all right, now there's introns and exons, and as we said, the introns are going to get cut out and the exons will be spliced together, all right, into the mRNA. So this is the mRNA now, the final one that's going to go out to the cytoplasm and be translated. Some other modifications that get made is there's going to be a guanine cap added to one end and a poly A tail, which is a whole bunch of adenines added to the other side. And so there's some other modifications. This guanine cap is going to help it dock on the ribosome in the correct place, and the poly A tail actually can help keep it from being eaten up by enzymes in the cytoplasm. So it's kind of a safety thing. All right, so this final mRNA is what's going to go out to the ribosome and be translated into a protein. Now, back to looking at mRNA modification. So once the pre-mRNA is made, and it can be spliced actually in a couple of different ways. And the protein unit, like this functional unit that does that, is called a spliceosome, which I think is a really cool name for a protein functional unit. So it's a spliceosome, and it's going to cut out the introns and splice together the exons. Now, something very interesting is that the DNA, generally we used to hear that one gene equals one protein or one polypeptide chain. And that was the original thought. And they used to think that humans had like 100,000 genes. Turns out that actually one gene can result in an mRNA, pre-mRNA transcript. So here's that mRNA, but can be spliced differently, that mRNA which can result in different proteins. So here's the one gene, and of course it'll have one pre-mRNA here, and then that mRNA is going to be modified by cutting out introns and splicing together exons. But sometimes what's considered an intron in one version, so here's the pre, and notice here the 4 is considered an intron. It gets cut out with the rest of this stuff. And that results in one particular sequence of mRNA nucleotides that will then be translated into a polypeptide. But there's another way it can also be spliced where the number 3 part gets cut out and the 4 is included. So the 4 in this one is an exon. And so this RNA splicing can happen in a couple of different ways. And that means that this one gene can result in actually a couple of different proteins. And so it turns out that because of this, for the ability to modify mRNA in different ways, it turns out that we actually ended up having a lot fewer genes, only 20,000 genes, but it's still why we can have so many more polypeptide chains than the number of genes that we have. So this is just another way that we can modify gene expression in eukaryotic organisms.